Human-dominated waterways contain thousands of chemicals, everything from heavy metals and ammonia to pesticides and PFAS. These chemicals can have varying negative impacts on ecosystems, making it difficult not only to assess the toxicity of a single compound, but also establish a stressor-casualty relationship when multiple stressors coexist. The In Situ Toxicity Identification Evaluation Technology, or iTIE for short, was designed to determine the presence of different chemical classes and to rank those chemical classes by the degree of stress they cause on ecosystems. It was developed by Alan Burton at the University of Michigan with funding support from CERTIP as a valuable field tool that is more efficient, user-friendly, cost-effective, ecologically realistic, and has fewer sampling errors than the traditional lab-based TIE method from the US EPA. Let's take a look at a graphic illustrating the broader iTIE system and how each component is related. For simplicity, we have divided it into three subsystems: water collection, iTIE sampling, and pumping subsystems. We can start by looking at the pore water sampling subsystem. Our pore water sampler, also referred to as the Trident, allows for water to be extracted from up to 150 centimeters below the sediment-water interface. As you can see, there are 6 probes on the Trident, each covered in a stainless steel mesh and a layer of glass beads to prevent sediment from entering the system during extraction. It also has a stopper plate that rests on the surface of the sediment when the Trident is inserted, which keeps surface water from being drawn down and mixed into our pore water samples. After water is collected from the site, it will enter the iTIE sampling subsystem. First, the water passes through an oxygen coil consisting of a small diameter oxygen tube inside a larger diameter water flow tube. As water flows through the larger tube, oxygen is gently diffused into the water. This is used to eliminate oxygen deficiency as a cause of lethality for test organisms. Once the water is properly oxygenated, the water flows past an overflow system to a manifold, where it is then diverted to 4 different iTIE units. Each unit consists of 2 parts, a resin chamber and an organism chamber. Site water first passes through the resin chamber, where the water slowly flows through an ion exchange resin matrix. Resin chambers can be loaded with different diagnostic resins, each utilizing physiochemical properties to pull specific contaminant classes out of site water. For instance, zeolite can remove ammonia, Chelex can remove heavy metals, C18 can remove nonpolar organics, and Oasis wax can remove PFAS molecules. Users can select different resins depending on the chemicals present at each site. After the water has been chemically fractionated in the resin chamber, it will slowly flow through the organism chamber, where various test organisms can be placed. Acute and chronic toxicity responses in those organisms can be observed and assessed. To date, we have successfully used freshwater daphnids, freshwater amphipods, mysid shrimp, and larval minnows in the iTIE system, though other test organisms can be used as well. Finally, the water is collected in sample bottles for additional water quality testing at the end of the run. Water movement through the system is fully driven by a series of peristaltic pumps. Peristaltic pumps can overcome gravity to draw site water up through the Trident sampler, through each iTIE component, and into the sample bottles while maintaining an even flow rate through the entire system. Currently, we are using Versa pumps manufactured by Ecotech Marine. These pumps can be programmed through the Mobius app, allowing for a high degree of precision and control. That is it for our overview of the iTIE system. In the next video, we will show how to set up the system and conduct a field deployment.